All right, should we go ahead and get started? 11 o'clock. Okay. All right, hi, my name is Ginny. I'm the relationship manager here at the Mother's Milk Bank of the Western Great Lakes. We're going to be hitting record. We will be recording this webinar today. Today's topic is advanced donor milk topics. My name is Ginny, as I mentioned. We are hosting this webinar in partnership with the Nutrition, Obesity, and Health Equity Research Group of University of Illinois at Chicago. And all participants from today's webinar will receive a certificate of attendance. We'll also be applying for CERPs for IBCLE, and we will also be applying for CECs from ALPP. That'll be available to the CLCs, ALCs, and ANLCs. At the end of the webinar, we will share a link to the post-webinar survey, and we appreciate any feedback that you can provide us at that time. Anybody who answers the survey will be entered into a raffle to win a $25 gift card and a Milk Bank WGL goodie bag. So today we're going to go into detail about the bioactive properties of milk. We're going to discuss the microbiome and review clinical indications for donor milk outside of the NICU. And at the end, we will have a Q&A session. So some of you have already submitted questions through the registration. We'll be including some of those at the end if they're not otherwise discussed in the presentation. And then I'd like to introduce today's presenters. Unfortunately, we are not joined today by Hannah Howell as planned. We hope we can introduce her to you at a future presentation. But with us, we do have Summer Kelly and Dr. Ann Eglash. They're both co-founders of the Milk Bank WGL. Summer is our executive director.
Grace. Thank you.
As a nurse, lactation consultant, childbirth educator, and researcher, Summer is passionate about the amazing bioactive properties in human milk. She holds a BSN from Rush University and an MS in biology from Northeastern Illinois University. In 2011, Summer helped develop the first
It's gone mute.
Ginny, you've been muted.
How long was I muted for?
Maybe 10 seconds or so.
Okay, so I'm going to just start Summer's bio again, if that's okay. So today we've got Summer Kelly and Dr. Ann Eglash. They're co-founders of Milk Bank WGL, and Summer is our executive director. She's been our executive director since 2015. As a nurse, lactation consultant, childbirth educator, and a researcher, Summer is passionate about the amazing bioactive properties in human milk. She holds a BSN from Rush University and an MS in biology from Northeastern Illinois University. In 2011, Summer helped develop the first donor human milk hospital program in a Chicago area level 3 neonatal intensive care unit. And in 2017, Summer and the research team here at Milk Bank WGL developed our first in-house microbiology lab called Lennon's Lab. And Dr. Ann Eglash, she is our medical director. She is an MD, IBCLC, and FABM. Dr. Eglash is a clinical professor with the University of Wisconsin School of Medicine and Public Health in the Department of Family and Community Medicine. In addition to family medicine, she has been practicing breastfeeding medicine since 1994. Dr. Eglash is a co-founder of the Academy of Breastfeeding Medicine and the medical director of the University of Wisconsin Breastfeeding Medicine Clinic. She has been published many peer-reviewed articles on breastfeeding medicine, has been the lead author in several Academy of Breastfeeding Medicine protocols, and has been an associate editor for Breastfeeding Medicine Journal. She co-hosts and produces a free bi-monthly breastfeeding medicine podcast series called the Breastfeeding Medicine Podcast. And she is the founder and president of the Institute for the Advancement of Breastfeeding and Lactation Education called IABLE. It's a nonprofit organization dedicated to the creation of breastfeeding knowledgeable medical systems and communities. So we're very happy to have both of these speakers here with us today. Nicole Geringer is our education manager. She will be available in the chat to monitor those questions or direct them to the Q&A at the end. And lastly, I'll introduce you to Marjorie Kirsten with the University of Illinois at Chicago. Marjorie is the project lead for the No-He Breastfeeding Equity Project. Thanks for joining us today, Marjorie.
Thank you so much for having me. So again, my name is Marjorie Kirsten. I use she, her pronouns, and I'm the project lead for the Nutrition, Obesity, Health Equity, and Food Justice Research Group at the University of Illinois at Chicago. And one project that we're working on right now is the Illinois State Physical Activity and Nutrition grant-funded breastfeeding equity project. So that was initially how we were connected with the milk bank folks and the wonderful work that they're doing. They were one of our grantees during our last grant cycle. So we're really excited to continue working with them and collaborating on these webinars. And if you're interested in learning more about our breastfeeding equity work, I can put the link to our website in the chat, and you're more than welcome to reach out to us. We have a variety of webinars throughout the year, and so we encourage you to join us if you're interested in learning more. Thanks so much.
Thank you, Marjorie. Thank you to everybody at NOHI. We really appreciate this partnership, and also Health Connect One for helping with the presentation. So thank you to both of those fantastic organizations. We are so excited to present today about advanced donor milk topics, and I hope you guys have your coffee or tea, because this is going to be really exciting. We get to really dive into the biology of human milk and donor milk science. And Ginny, can you just let me know if the screen looks okay? You can see the full screen and not all of my Zoom controls.
Yep, it looks perfect.
Okay, great. Thanks so much. So to start, we're going to kind of talk about two different models for how to interpret, understand, and think about lactation science and medical science. So on one hand, we have the medical model, which a lot of us are familiar with. There's a lot of nurses on the call today. There are peer counselors and people who work at WIC clinics and physicians. So we're all very immersed in the medical model. And this model is human focused. There's a strong reliance on published research. And there's an emphasis on the current body of research that we have right now. So that's kind of the medical model that we work on, very research and evidence-based. The biological model is a little bit different. So the biological model, let me move my stuff here. So the biological model really looks at human lactation in the context of hundreds of millions of years of mammalian evolution. So if you look at this phylogeny or family tree over here, you can see, and this is kind of starting at the bottom and going up. So I know that some phylogenies have, oh, let me pull this up really quick. Okay, there we go. So I know that some phylogenies have, they kind of go from left to right, but this is a tree coming up from the ground. So you can see that at the very bottom of the tree, that was a long time ago, 360 million years ago is when the tree starts, and we start splitting off from modern day birds and reptiles 300 million years ago. So when we follow the tree on the left to where the mammals are, we can see that hundreds of millions of years ago, we started developing the genes and the proteins and the structures to lactate. So this is a very ancient process, and this is really encompasses the biological model. We're thinking about all of these hundreds of millions of years of evolution. We understand that there's a basic and fundamental understanding that human milk is the healthiest choice, with rare exception. This is just incorporated into the biological model. We know that human milk is the healthiest choice. And we also know that we don't fully understand human milk's components, mechanisms, interactions, nor do we fully understand the impact of formula. So in the biological model, we do consider current research, but we also think about what we don't know, how complex human milk is, all of the hormonal interactions and the interactions with growth factors and cell signaling, which we're going to talk about today. So here's another beautiful phylogeny. I love biology because it's so colorful. So you can see that mammals are very, very diverse. We have, a tiny little bat that can fit on your finger, and a huge blue whale that roams the ocean, the biggest animal that we think has ever lived on Earth. So we are just a very diverse group of animals, and our lactation strategies reflect that. So our lactation strategies are shaped by natural selection, by all of these hundreds of millions of years of evolution. And we know that there's significant differences in the milk from all different types of mammals. So if you're a big blue whale swimming around in the ocean, you're going to want to provide really fatty milk to your babies, to your calves, so that they can quickly deposit blubber so that they're nice and warm in the ocean. If you're a human or a primate, you're going to put a lot of polyunsaturated fatty acids into your milk for brain development and for cell membrane integrity and such. So there's a lot of differences between all these different types of groups of mammals and the milk that we have. And we know that there are significant differences between human milk and cow milk. Cows get up right away after they're born. So their milk is full of protein for really strong bones and for muscle development. And humans are kind of helpless when they're born, right? So moms and parents carry them around, and their milk is very different. So one word that we're going to come to a lot today is homeostasis. Homeostasis is the natural order, natural balance. And this is really important in the microbiome, which is the focus of our talk today. So we're going to kind of focus on two main components in human milk. One is a carbohydrate, which is just a bunch of monosaccharides stuck together. And that is called a human milk oligosaccharide. So it's a short chain sugar structure that has a ton of activity in the microbiome. And the other structure is protein called IgA, or immunoglobulin A. So we're going to learn about secretory IgA as well. So one carbohydrate and one protein. So the protein that we're going to learn about is an antibody. And antibodies are immunoglobulin proteins that bind to antigen in our bodies to keep us healthy and safe. So an antigen is any type of foreign particle. It can come from a virus, a bacteria, even pollen or pet dander. So if you're like me and you have allergies, you know all about antigens because that's what induces an allergic reaction. Antigens can be harmful to our body because they can not only induce allergies, but inflammation as well. And sometimes our body would recognize these antigens as foreign invaders and launch a full-blown immune response. So that causes symptoms and problems that we aren't really, especially for a newborn baby, that we would try to avoid. So this antibody here is shaped like a Y. And that is recognizing this guy right here, this E. coli. You can see that he's a dangerous bacteria because his eyes are furrowed and he looks really angry. So he is about to cause some trouble. He wants to get inside one of our babies and attach to the baby's cell lining to make the baby sick. So the good thing is that the antibody's blocking him. Do you guys see how the antibody's attaching to his arm or his flagella or cilia, whatever appendage or protein adhesion he has to stick to our baby's stomach? That antibody's binding to it and neutralizing that bacteria so he can't attach to the baby's stomach. So we know that there's 5 different classes of antibodies. And if you work in a clinic or hospital, you sometimes come across these. So if a baby is born to a mom with hepatitis B, we immediately give hepatitis B immunoglobulin. So that's an IgG. That's this first antibody here. Very important antibody, very active in the blood and in tissue. We also have IgD, IgE, which gives us allergies, some of us. Secretory IGA, which is a dimeric structure. So it's 2 antibodies that are stuck together. And look at IgM. It's this huge pentameter with 5 antibodies stuck together. So that's a lot of surface area for that antibody group to bind to. So that's really good at being one of the first responders to infection and inflammation in our body. But we're going to focus on secretory IgA, the main dominant antibody in human milk. And it's really cool because it's made for human milk. Like it's made to protect our mucosal surfaces, our GI system, our respiratory tract. So if we look here, these two antibodies, these two IgA antibodies are stuck together with a J chain, which is a little protein. And then we have this secretory component that wraps around it. This is really important because it prevents IgA from being metabolized and broken down in our stomach, and which is really good so that can maintain its anti-infective properties and its immunomodulating properties in our baby's stomachs because it's not degraded by digestion. So this is a little image here showing this is the cell membrane for the baby's stomach. So if you think about the baby's stomach, small intestine, the entire GI system, really the small intestine is what we think about when we talk about the gut and infant health. So when we think about this, we can see that the cells, just like all the cells in our body, have this cell membrane, this double phospholipid bilayer membrane here. And there's proteins, the integral proteins that are in there. And we have these little receptor sites that are made up of oligosaccharides. And these are really important. There's a lot of interaction and communication between these cells in our babies and the milk and the formula and all the pathogens and bacteria. Everything that comes in through the baby's mouth and into the GI system interacts with these cells. So we can see that the bad E. coli is trying to attach. So you can see he's got some little flagella down there, some little legs. So he's trying to attach to our baby's cell in the GI system to make our baby sick. But luckily, mom just breastfed baby, or the baby just got a donor milk feeding in the NICU. So there is secretory IgA that is protecting this baby cell from being attached. This is called neutralization. This is a major role of secretory IgA and also human milk oligosaccharides. So they do the exact same thing. It's really cool. So both the HMO carbohydrates or sugars, as well as the IgA protein or antibody, can protect our babies from pathogenic bacteria. Another cool thing that they can do is agglutinate the bacteria. That's where they attach to the bacteria and then just completely mob him so that he's just covered in antibodies and he can't actually make our baby sick. So this is another way that antibodies protect our babies. So that was just kind of a very simplified view of how antibodies protect our babies. Now we're going to drill down and kind of open our minds to the microbiome. And I say that because we're starting to change the way that we think about the microbiome. So if you look at this image, and this is my favorite paper about the microbiome, this is a 2020 paper by Berg's group. And they have a new definition of the microbiome. It's based on an older version from 1988, and then they just kind of expanded on that. So when we think about the old way that we think about the microbiome, let's look at the top of this image right here. It's the separation model, where we kind of separate and put pathogens into distinct groups or buckets based on how we categorize them. So maybe the E. coli that we were just talking about, maybe that is a pathogen, but maybe sometimes he could be a commensal. So, you know, this doesn't really work all the time because bacteria and the microbial populations that are inside of the microbiome are dynamic. They change, they interact with each other, and there's opportunistic pathogens as well. Maybe the E. coli just hangs out and doesn't cause any trouble until the conditions are just right, and then they're going to wreak havoc and cause a lot of problems. So this separation model doesn't really work. It does, and this is how we are geared to put things into categories. Biologists love doing that. So we've been doing that for a long time. But let's become more holistic. Let's think about the microbiome as integrated within us. We are a super organism. So I'm not just summer. I'm summer plus, you know, a trillion bacteria, yeast, fungi, all types of different organisms that make up my microbiome. I'm like a universe walking around with all sorts of cool dynamic populations living inside of me. And that's how all of us are. So we are our microbiome. We are one with our microbiome. This is not a separate invader that's causing problems. This is integrated into our bodies. bodies. So this is a holobiont evolution perspective and the holistic approach. One health, everything is in balance. So this is homeostasis. Okay, I think I brainwashed you guys enough about this microbiome model. So we'll move on with this really as a foundation for what we're talking about today. So this is a micro ecosystem that's integrated inside of us. And there's a theater of activity. The bacteria that live inside of us, they do essential functions that are required for our health. They help us with metabolism. They break down things that we are unable to break down. They recycle nutrients to give them back to us. They protect our mucosal barrier. They're like almost like security guards, you know, monitoring everything and making sure that pathogenic bacteria don't come too close. And they communicate directly with the cells in our baby's bodies. With cell signaling, they create change. They tell the cells to do certain things, to grow, to proliferate, to become anti-inflammatory, to become pro-inflammatory. There's all sorts of cool interactions. So this is homeostasis. It comes back to basic biology. It comes back to the natural order of a parent breastfeeding their child. So we can see here that this is a give and take. It's a two-way communication, three-way, four-way, because of all the different types of populations that we're talking about here. So mom has, and the parent has, an entire ecosystem of bacteria and abiotic and biotic factors living in the milk microbiome. So the milk itself has bacteria in it, which is normal, has all sorts of abiotic factors. So just to go back really quick to talk about biotic and abiotic, biotic are living organisms. So the bacteria, the fungi that live inside of our milk and inside of our babies. And then the abiotic factors are things like pH, fluid dynamics, the things that are not living, food that we eat, medications that we take, those are abiotic factors. So the breastfeeding microbiome has living bacteria to help seed the baby's microbiome. So parent is sharing their bacteria with the baby. This also provides human milk oligosaccharides, which serve as a food source for the bacteria and also can neutralize pathogens like we saw. The secretory IGA has the same two things and promotes homeostasis in many different ways. There's also tons of other factors, growth factors, hormones, cytokines that communicate directly with the cells in baby's stomach. And these are only found in human milk. And then we can have communication from the baby to mom. So this is retrograde flow. A little bit of a vacuum can pull some saliva from baby's mouth into the breast. This may contain signals, hormones, antigens that can program and communicate with mom's microbiome. How cool is that? So when I first became a lactation consultant, we learned that human milk is sterile, and sometimes there may be some skin flora that comes off, into the milk. And now we know, this is a study from 2011, that is not true, that human milk has dynamic populations of bacteria. So this image here, when we talk about the microbiome, we get to look at all these really colorful research studies. So if you look at S1, that's subject one. So this is one mom and her milk, and you can see all the different types of bacterial communities that live there at 3 different time points. So not only is milk different from person to person, but it's also different based on the timing or the day or, the mom's use of antibiotics or any number of factors. A ton of factors influence the maternal human milk microbiome. So here's some of those factors. So for biotic factors, it is the microbial communities that are living inside of the breast and in the milk compartment. We have abiotic factors like temperature, all sorts of things. We have macronutrients and micronutrients, and of course, tons of bioactive factors. And these all influence the human milk microbiome. And then there's clinical factors. For those of you that work in the clinic or the hospital, the things that we do and the treatments that we prescribe or administer have an impact on the maternal microbiome. So this can, but like antibiotics is a classic example, even maternal diet possibly and structural breast issues like edema or engorgement. All of these can impact the human milk microbiome. All right, now we're going to dive into the baby's microbiome. This is going to be really fun. So let's get ready to shrink down, and we're going to jump right into our baby's small intestine. Are you guys ready? Okay, so the pink layer on the bottom is the baby's stomach lining, or the small intestine lining. So this is the baby's epithelial lining. And then in the kind of space above, that's the lumen, or the center of the intestine, where the milk goes, where the food goes through. So you can see the E. coli's here. And by the way, not all E. coli are bad. I'm really picking on this guy, but he's just a representative sample of a pathogenic bacteria. We also have a representative sample of a probacteria, or a commensal or good bacteria that keeps everything in balance. And that is Bifidobacterium longum, subspecies infantis. So we can just call him Bifido. So this guy is a good guy. He is going to keep everything in homeostasis and balance. And look, this baby is in the NICU, and this baby just had some donor milk. So there's oligosaccharides that are in the intestinal lumen. We have human milk secretory IGA. And there's a lot of abiotic factors that are in play right now, like the flow, the osmolality, which is like a density of the feedings and the fluid. We also have the mucus, the surface receptors in the baby's stomach, all sorts of things. This is a very complex ecosystem. So we're going to pare it down and look at Bifido, our buddy, who's keeping our baby safe. And he's like, oh my gosh, a human milk oligosaccharide. I love these. This is like deep dish pizza, or for our friends in Wisconsin, it's like a brat fresh off of the grill. Best food ever. So he's going to chow down and eat this human milk oligosaccharide that was in the donor milk feeding. Oh my gosh, there's more. So the baby just got a little bolus there of some feeding. So Bifido is like, man, this place is awesome. I'm going to, settle down and have a family. And this is great because this is a competitive exclusion. So Bifido is taking over the landscape of the small intestine. There's no room for E. coli. He's coming in. Now there's nowhere to go. In addition to that, the human milk oligosaccharides neutralize this guy and prevent him from attaching to our baby's stomach. Secretory IGA can do the same thing. So the secretory IGA here is neutralizing this guy. It can also do this really cool thing called enchaining or agglutination. That's where the secretory IGA forms like a chain of bacteria. There's some cross-linking that happens. And now these guys can't share genes. They cannot do something called horizontal gene transfer, where they share genes for antibiotic resistance or virulence to make them more pathogenic. So this is a good thing. Really good that we have access to donor milk. And there's new research that shows that the Bifido may like to eat part of the IGA too. So we know that they love human milk oligosaccharides. That's their favorite food, but they can munch on some secretory IGA perhaps too, especially the carbohydrate motif near the hinge region of the IGA. So that's some really cool new research. And I think a really good question is, how does the stomach and the baby and everybody, the bacteria, the IGA, how does this all work together to keep the good bacteria close and clear the bad bacteria? We haven't quite figured that out yet, but this study is one of my favorite studies about secretory IGA. It's fantastic. So this should be on your summer reading list. And this is a study from 2011. And I love this image because it does such a great job of trying to explain this really complex topic. So these are the same epithelial lining, right, that we saw in the baby's stomach. So you can see the IGA, and it's communicating with the blue bacteria, which is the good bacteria. And you can see here some cell signaling going on for under letter A. So the plus sign and the minus sign shows what the cell signaling is doing. It's promoting something, or it's decreasing something. It's decreasing in inflammation, and it's promoting gap junctions. It can do all sorts of stuff. It can tell the cell what to do. It's so cool. And we can see over in C that really shows how the secretory IGA can clear the bad bacteria and keep the good bacteria close to form that positive biofilm. This is another image, electron scanning microscope image from that same study. So in the top, they show salmonella just hanging around in some PBS or some water or something like that. So salmonella just minding its own business. And then for B, the only difference is salmonella minding its own business with secretory IGA. Look what happens to that salmonella. Gets totally messed up. So the secretory IGA, and we don't know exactly how all of this works, but it's really cool because we can see it happening. The secretory IGA, the same secretory IGA that's in mom's milk and donor milk, does this to bacteria. It totally messes it up. You can see that cross-leaking, the enchaining, the agglutination. It's just a big mob of dead or dying bacteria. This is a great study. This is a row here from 2014. I absolutely love this study. So I won't go into all the details. I'll just kind of give high level highlights, and this should also be on your reading list. What these researchers did was really brilliant. I love the design of the study. They created mice that either gave IgA or were unable to give IgA, and little baby mice or pups that were able to make their own IgA or unable to make their own IgA. So they came up with 4 research groups. And what they found was the ability to block pathogenic bacteria, which you can see in the bottom graph, the aerobic colony forming units, is that it was based on the maternal genotype, her ability to put IgA into the milk. If she was able to, then you had almost no bacteria. And you can see those clean plates under B. See the clean plates? So that's when mom is able to put IgA into her milk. When she's unable to do that, you can see those dirty plates. You can see all that bacteria. So IgA protects baby pups, baby mice against aerobic bacteria. Now, this is my favorite part of the study. So this shows weanlings on the left, that ring with all the different colors of bacteria, and adult mice on the right with all those different colors of bacteria. Look at C. So that's called a coordinate analysis, and that groups the bacteria based on certain factors. What I really love about this study is it shows that the difference between the bacterial groups that you see in the microbiome is more pronounced in adult mice. And that is really mind-blowing because this shows a lifelong impact of the microbiome. So if we look at this chart, we can see the yellow and green are overlapping a little bit, and they're kind of clustering together. Those are the weanling mice or the baby mice. So they're different, but they're kind of similar. And then if you look at the blue group and the gray group, those are the adult mice, 2 totally distinct separate groups of the microbiome. So that shows that the ability to give babies IgA, at least for mice, leads to lifelong benefits and immune programming. This is another study that they had that kind of explained it, their complex study to a general audience. And I love this because if you look on the left, you can see this mom is breastfeeding, and you can see the IgA is going into the baby's system. When you look at the intestine, it's beautiful. Look at that mucosal barrier, and all the bacteria are being kept in check by the secretory IgA. Over on the right, this mom is formula feeding. There's no secretory IgA. And unfortunately, the bacteria are able to breach that mucosal barrier and go in and start damaging the stomach cells, which can lead to inflammation, possibly necrotizing enterocolitis. And there's some new research about that. So this is the research that shows that secretory IgA may protect against necrotizing enterocolitis. And they found that IgA bound to bacteria is correlated with lower risk of NEC. So that's, you can add this to your reading list as well. Really important for NICU providers to understand that the components in human milk have an impact on reducing the risk of necrotizing enterocolitis. So what about donor milk? We don't always have access to mom's milk in the NICU. So we have donor milk here, and then we're going to compare that to formula because mom's milk is always a gold standard. We need to invest a lot of resources into making sure that moms can reach their breastfeeding goals, especially when they're in the NICU. So if we don't have access to mom's milk, we can use donor milk, which has 70% of the IgA retained for all of those microbiome effects. And this is specific to the, specific to the microbiome. So we will provide slides to all attendees after. So Nicole will email you guys all these slides so you can reference these in the future. And I'm going to close with a recent study. I was just talking to Ann the other day, Dr. Ann Eglash, and I said, we need a study that shows how donor milk and formula and mom's milk are different related to the microbiome. I said, if you did a PCA or a coordinate analysis, you would see the donor milk and the mom's milk grouping together and formula would be separate. And that's already been done. And that's exactly what it showed. So, I mean, if you understand biology in the microbiome, you can predict what the research will show. It's really cool. So this is a coordinate analysis or an RDA looking at donor milk and mom milk. There's some overlap, but look when you throw formula into the group. There's a ton of overlap between donor milk and mom's milk, and then the formula is off to the side. Exactly what I would have expected. You can also see here, these are those beautiful color charts with the bacteria, mom's milk, donor milk, and formula. You can just see the differences. And then when you run a PCA and you look at the operational taxonomical units, which are just like the genus level, wow, look at this. I mean, the donor milk and the mom's milk are creating similar microbial communities, and the formula communities are not similar. They are in a separate coordinate. This is even more important. This is the metabolic functions of the microbial communities within the microbiome. Look at the overlap between donor human milk and mom's milk, and formula is way off to the side. It is very different, and this really brings that home. Which group do you guys think, if you can't read the words, you can see the formula group, right? You can just look at this and see how different it is. It's the blue dots in the middle. So look at the red dots and the gray dots and how they line up, and look at the blue dots. These are box and whisker plots. So this shows the median and the quartiles. Unbelievable. My mind is blown right now. I hope you guys are understanding how important this is. Donor human milk, when you don't have access to mom's milk, helps promote a healthy microbiome that is more similar to the mom's milk fed group of infants. So I'm going to close now just showing the breastfed infant microbiome, homeostasis. This is what happens when we don't have access to human milk and we can only give formula. And this is what happens when we can use restoration ecology to reshape and remodel the infant microbiome with donor milk and possibly with probiotics. So I'll let the AAP work out the probiotic issue. And now I will turn it over to Ann to finish our discussion.
Thank you. That was so excellent, Summer. And we're going to talk more about the microbiome. And we have some great research coming out of Canada with Dr. Azad showing the importance of the microbiome, particularly for obesity and also blood pressure. There's just a number of things that are coming out that are very exciting. So I'm just going to share my screen here. Can you see my screen? Anyone?
Looks great. And you can go a little bit over because we actually do have until 12:30. So please, take your time with your discussion.
Okay, yeah, I have to go to work actually, so I might have to leave a little before then. Okay, so we're going to talk about pasteurized donor human milk for well newborns. I'm just going to actually, give me 1 second. I just want to grab my views here. Okay. So this is kind of a relatively new phenomenon that's happening in this country using, and when I say donor milk during this talk, I'm actually referring to pasteurized donor human milk, but just to save all those words, I'm just going to say donor milk. So first, we're going to talk about the frequency of formula supplementation in US hospitals right now and the impact that it has on the duration of breastfeeding. We'll talk about the impact of donor milk use in level 1 nurseries on breastfeeding duration. Then we'll talk about common reasons why donor milk is being used in level 1 nurseries. And again, when I talk about level 1 nurseries, I'm talking about the well baby floor. We'll talk about the impact of donor milk for supplementation for those who are at risk for asymptomatic hypoglycemia. So I'm not going to talk about donor milk use for infants who have symptoms of hypoglycemia. They're automatically, usually going to be going to the NICU and oftentimes receiving IV glucose. So we're not going to talk about that group. We'll talk about just how one bottle of formula can impact the gut microbiome, which Summer just went over the importance of the microbiome. We'll talk about the risks of formula supplementation for infants whose lactating parents intend to breast or chest feed, and just some other issues regarding using donor milk on the floor, particularly among attitudes and concerns by parents and nurses who have been involved with using donor milk. So this is our 2020 Center for Disease Control breastfeeding report card that shows where we are in terms of Healthy People 2020, and we've reached many of our goals, not exactly all of them. But if you actually look at the percent of breastfed newborns who receive formula in the first 2 days, it's 19.2. And that, we'll never be without supplementation in the hospital outside of mother's own milk, because we know that as time goes on, we're seeing more and more individuals who never would have been pregnant, 20 years ago. With all the advances in fertility, we're seeing more and more people who have risk factors for a delay in lactation, meaning it takes a while for their milk to come in. So we're talking about individuals with higher BMIs, older age, other complications that are related to preeclampsia, gestational hypertension, et cetera, hyperandrogenism. So there are lots of people who are giving birth who have several maternal conditions that increase that risk of a delay in lactation. And so they're going to need supplementation. So this is never going to be zero. This is not a failure, so to speak. But the question is, can we use donor milk instead of formula in these situations? We know from evidence that's been studied for quite a while now that the more we use formula in those first few days, the greater risk that there is of weaning by 2 months of age. And in fact, so this was a study that's oftentimes cited by various papers and talks showing this. Carolyn Chan, she showed this in 2014, but, and I didn't include a slide on Valerie Flayerman's research, which I think was published in 2020 or 2019, but she did really interesting research. She wanted to find out if giving a little bit of formula, like 10 ml with every feeding the first couple of days, would actually prolong breastfeeding. And her theory was that when someone's breastfeeding, when mothers are breastfeeding and their babies are really losing a lot of weight, like they're down to 11, 12%, and everyone's concerned, and the doctor says, oh, you need to supplement your baby, give your baby like an ounce and a half of formula after breastfeeding. What her thought was, well, when you ask women to supplement that much, that may actually be more harmful to the duration of breastfeeding than if you just give a little bit for that support, waiting for secretory activation to come in, and then they can fully breastfeed and you can just drop the formula. Well, what her studies actually found were that so we're talking about women who had very high intentions to breastfeed in both groups, so they were randomized, right? So those who received that little bit of formula in the first several days, in the first couple of days, I should say, were much less likely to be breastfeeding at a week, even though they had high intention. So there's something about giving formula early that consistently shows that there's a decreased likelihood of breastfeeding at the time of hospital discharge and duration of breastfeeding after they leave. The most common use of formula is really for inadequate intake early postpartum. We'll talk about that. And particularly hypoglycemia is a major reason for supplementation early. So this was a study that was published just last year, looking at the impact of exclusive breastfeeding at 6 months with the use of donor milk in the level 1 nursery. So at the University of Florida in Jacksonville, they began using donor milk on the floor in 2015, and they would allow anyone to choose donor milk if the baby needed to be supplemented. So the mothers could choose pasteurized donor human milk, they could choose formula, and if they chose the donor milk, they had to sign a consent. And this was a baby-friendly hospital. So they had all the systems in place to be, to optimally feed babies. So they did a chart review of about 3,000 infants and found that about 122 of these infants were supplemented. And then they looked more closely and they found that 73 of these 122 received formula and 49 received donor milk. And so they were interested in finding out, looking in contacting them 6 months later to find out how long they ended up exclusively breastfeeding. Unfortunately, 50 dyads were lost to follow-up. But anyway, among those who were asked about whether or not they were exclusively breastfeeding, they found that among those who chose donor milk, that you can see there's a really high rate of exclusive breastfeeding at 6 months or beyond, like close to 60%. Whereas those who chose formula had a very small percent of those who went on to exclusively breastfeed for the first 6 months. Whereas breastfeeding for less than a month was like the vast majority of babies who chose, of those dyads who chose formula, and a much smaller percent of those who chose donor milk early. Now, granted, this was not a randomized controlled trial. These families could choose for themselves what they wanted, and that intention may have had an impact on this. But nevertheless, this is what they found, which is quite striking. This was a study that looked at the use of donor milk in a particular region of the United States. So these authors, they sent out surveys to hospitals, all hospitals in Massachusetts, and also to hospitals that were served by the Mother's Milk Bank Northeast, which is in New England. And they wanted to see when they began using donor milk on the level 1 nursery on the well baby floor. And back in between 2008, 2013, it wasn't really very common to be using donor milk, but then the use of donor milk on the floor really shot up significantly after 2013. And so then another group of authors decided to look at the policies among these level 1 nurseries who are using this donor milk. And so they looked at the eligibility criteria just to see like who used what eligibility criteria. And for the most part, the majority of them used medical conditions of the infant. If a parent requested it, they allowed it. Poor feeding at the breast was one of the eligibility criteria. Just a few had criteria for the mother, and then multiple gestation was another one. But I want to talk a little bit right now about the medical condition of the infant, like what were those eligibility criteria. In addition, I just want to mention that all these hospitals, except for 2, required written consent in order to use the donor milk. One hospital policy said that it could be written or verbal, and one actually didn't specify whether it had to be written or verbal. When you look at the medical eligibility for infants, the use of donor milk for the floor primarily has been used for hypoglycemia and hyperbiliary with phototherapy. So those were like the top 2 reasons for using it. Excessive weight loss and birth weight, so particularly low birth weight or IUGR, would be reasons for using it for automatic supplementation. And then gestational age and hydration. I was a little surprised to see gestational age as low down, because in our hospital that has been baby friendly for, gosh, I think since 2005, we have a routine policy for providing supplementation on the floor, for babies who are like 35 to 38 weeks, used to be 35 to 37 weeks. And for a while it was formula and obviously expressed breast milk. And then a couple of years ago, we started using donor human milk on the floor for that group as well. And it would seem to me that that would be a common reason in many other hospitals as well. And then hydration is a reason as well, but oftentimes insufficient hydration, which would be a weight loss of like 11, 12%, is not really going to be identified until after they leave the hospital, if they leave on day 1 to 2. The eligibility criteria for mom, again, most of the hospitals didn't really have eligibility criteria for the moms. About 3 hospitals used maternal infection as a reason. And then 1 hospital for each of these was identified using these reasons: breast infection, history of breast surgery, chronic infection, HIV, which is interesting because it seems to me that a lot of hospitals would choose to use donor milk in those who had the intention breastfeeding, and the whole HIV topic is, that's a whole nother topic. And there definitely are people who choose to breastfeed who are living with HIV, who have negative titers noted, no RNA load, but that's another topic. And then nipple pain and diabetes these are other reasons, and just like a few hospitals. So most hospitals didn't really have eligibility criteria for the moms for using donor milk. So the number one reason for hospital supplementation for well newborns tends to be hypoglycemia. And really it's very common, like 15% of term and late preterm infants have transient neonatal hypoglycemia, which is usually not going to be symptomatic. And of course, it's going to depend on the definition of hypoglycemia, which is very nuanced and a gray area for sure. It's not like it's not clean-cut definition. I want to mention that the Academy of Breastfeeding Medicine updated their hypoglycemia protocol in 2021. You can see any of their protocols for free at bfmed.org. And so this is a really nice protocol. This was very well done. And this is just one screen capture of their protocol. And you can see that for infants who are at risk, which is in a separate, they have a whole other table with the risks, infants who are at risk for hypoglycemia who should be routinely screened. They do recommend early breastfeeding and then supplemental feeds within an hour. And they include pasteurized donor human milk as one of the options for supplemental feeding. So that was exciting to see they saw that as one reasonable option. So I want to share this study that was published recently, just a couple months ago from Texas. So this was a hospital that wanted to see what would happen if they supplemented with pasteurized donor human milk for asymptomatic neonatal hypoglycemia. So what they found is that their routine protocol was that they would use the 40% dextrose gel and continue breastfeeding or have the parent express colostrum or whatever milk they could and give that to the baby as well to correct asymptomatic hypoglycemia. But they found that just by doing this, the protocol didn't really reduce their admission to NICUs. So what they were doing is they did skin to skin for the first few hours, as any baby-friendly hospital would do. And then they would make sure that feeding occurred within the first hour, and if not, would have the parent express colostrum and give that to the baby and check a blood sugar at 2 hours. And if the baby didn't have any symptoms, but the blood sugar was between 26 and 45, they were instructed, the nurses were instructed to give the buccal dextrose gel and then continue breastfeeding or taking express breast milk. And then we check the blood sugar again in 30 to 60 minutes. And if the sugar was still low, do the gel again and continue feeding. And then if it was still low the third time, just transfer the baby to the NICU. And they found that this kept happening, that even though they were using the dextrose and breastfeeding, the babies oftentimes went to the NICU. So they thought, well, maybe this is because the amount of colostrum and breast milk is just really low, because it is really low, right? And it's going to depend on whether mom is going to have a delay in lactation, whether she has insufficient glandular tissue. There's so many different reasons why colostrum can be low in the first 24 hours. And in primate, for sure, they're going to have less milk than a multip. So they thought, well, maybe what they could do is actually supplement with donor milk to increase that feeding volume along with the dextrose gel. And so what they did instead is their new protocol was to do skin to skin for the first few hours, and then make sure that there is feeding in the first hour naturally with either breastfeeding or expressed milk colostrum. And we check the babies for their first sugar at 90 minutes. And again, these are babies who are at risk. These are not babies who are automatically known to be low blood sugar. They're just at risk. And so if they were found, if they were asymptomatic and their blood sugar was 26 to 45, they were given the dextrose, they were told to continue breastfeeding or to express their milk, and they were given 10 ml of either pasteurized donor human milk or formula. Then they would check the blood sugar again in 30 to 60 minutes. And then if it was still low, do the same thing, give the dextrose gel again, breastfeed, and then add another 10 ml of donor milk or formula. And then if it's still under 45, then transfer to the NICU. So what they basically found is that the NICU admission rates for the asymptomatic hypoglycemia among those at risk decreased from 16% to 6% using donor milk. And among parents with the intention to breastfeed, they had an increase in exclusive breastfeeding rates from discharge from 33 to 55%. So they were able to not use formula, increase those exclusive breastfeeding rates, and keep these babies out of the NICU. And if you look at the mean dextrose change, so basically what happened is that if they had that hypoglycemic episode where they were asymptomatic, they were given the dextrose gel, and if they were continuing to breastfeed and just given the gel, their blood sugar would go up about 11.2. And if they were given formula, given the dextrose gel and formula, it went up to 12.9. So not really a whole lot of difference. And if they were breastfed and formula, interestingly, it went up 6.6, which was kind of not sure why. But then if they had them breastfeed and gave donor milk, it went up significantly, like in a major way. So there wasn't really a good explanation for these differences, but the important thing is that donor milk is not inferior, and in fact is probably superior. And it might be that, who knows, it might be that there was more limitations on the amount of formula that was given. Maybe babies don't like formula, maybe they spit it up, we don't really know. But nevertheless, donor human milk is definitely an option for supporting babies who are at risk for hypoglycemia. So what happens if formula is used? Well, so here we are trying to prove ourselves as something that's a natural product, right? Like this is normative feeding, is to give breast milk, and fine, this is pasteurized and it comes from donors, but it is natural, right? And so here we are trying to prove that this is okay as compared to formula. So shouldn't we be talking about, like, is formula really a problem? Well, it really is, and we'll talk about this. So we know from studies, which I already talked about, that when formula is given to families early postpartum, there's a lower rate of exclusive breastfeeding at discharge, even when there's strong intention, as I mentioned in the Flaterman study. There's a higher risk of cow's milk allergy, which we'll talk about. There's a degree of parental discouragement, and there's also a change in the infant microbiome with just one bottle. So there are 3 really strong studies that have come out in the last, I guess, really since 2012, that have shown pretty definitively that when you use cow's milk-based formulas in those first couple of days for infants who are then going to go on to exclusively breastfeed, you are really increasing the risk of cow's milk allergy in that population. So the first study that came out on this was in 2012 that showed a 1.75 times increased risk of cow's milk allergy by the age of 1 to 2 in infants who received just a little bit of cow's milk formula in the first couple of days, and then went on to exclusively breastfeed for the first 6 months. This was actually really much more profound in a study that was done in Ireland among 55 children who had cow's milk allergy. They found that giving cow's milk-based formula in the first 1 or 2 days increased risk by 7 times. And then there was a cohort study that was done in Japan that was published in JAMA Pediatrics a couple of years ago, where they actually intentionally gave formula to babies in the first couple of days and compared the cow's milk-based formula with hydrolyzed formula, where the proteins are chopped up and not identified as that cow's milk protein. And so they found that those who got the cow's milk-based formula actually had the higher risk of cow's milk allergy as compared to those who got the hydrolyzed formula. I think this has been compelling enough for us to not be giving cow's milk formula to infants where there's an intention of breastfeeding, and that if there needs to be formula and there's not the use of pasteurized donor human milk, that really hydrolyzed formulas or elemental formulas should be given for supplementation. And so one thing, so there's also this very exciting research that's coming out of Canada. So if any of you follow Dr. Megan Azad, she is the deputy director of the child cohort study. This is a large study that's a prospective study where they enrolled about 3,495 dyads in 2009. And I think they're continuing to add dyads, but they're just following individuals over time. Whoops, I think someone needs to be muted. And so as part of this cohort, 1,087 infants were followed to look at the risk of obesity and changes in microbiome. And so that's one of the main goals of this cohort study, is to look at the difference in the microbiome and to correlate the differences in the gut microbiome of the infant according to feeding with outcomes later, and wondering if the difference in microbiome is mediating some of the differences that we see in health in children regarding things like blood pressure and weight and high cholesterol and a number of other factors. And so what they found is that there is this dose-dependent increase in obesity with less breastfeeding. So the less breastfeeding they have, the higher risk of obesity. And they found that this was specifically correlates with differences in gut microbiota. So so basically in the infant gut, and I think Summer alluded to this, the gut microbiome in a baby who's breastfed is much more narrow. There's a lot more bifidobacteria and enterobacteriaceae, but there's in babies who are formula fed, there's like a really much wider range, a lot more variability in bacteria in the gut. So what they found, when they looked at this, they found that the mothers who said that they exclusively breastfed, where they didn't get formula, they had the most pure microbiome. So then the researchers looked at their hospital records and wanted to see, now, did they really all did they get any kind of formula whatsoever? And they found a small number of these dyads where mom said, oh yeah, we're exclusively breastfeeding. A small number actually got like 1 or 2 bottles of formula. And they actually found just in that group that there was less abundance of bifidobacteria in those infants' guts as compared to the infant gut of those who never got that 1 bottle. They didn't find that those babies who got 1 bottle had higher obesity rates, but there was a difference in microbiome. So we're messing with something that we need to be paying closer attention to, because again, if we actually start looking at this research and finding that the gut microbiome is so correlated with these other health outcomes, we want to really cherish it and worship it and protect it, and not add something that's artificial that's really going to change that. I have a couple other things I want to talk about, just like perspectives on choice of supplementation. So this was a study that looked at mothers of term infants and wanted to know what their perspectives were of using pasteurized donor human milk. So they interviewed 24 mothers of term newborns who gave birth, and they were still postpartum in the hospital in 2016. 75% of these mothers were exclusively breastfeeding in the hospital, even though like 80% had planned on exclusively breastfeeding until about 3 months. And that's because some were already needing to be supplemented. So they were asked, if you could choose your supplementation method, what would you choose? Would you choose donor milk or would you choose formula? And again, in each of those groups, 80% were planning to exclusively breastfeed until 3 months. There weren't really any differences in terms of race, ethnicity, age, socioeconomic status between these groups. So 58% again chose donor milk. 42% said that they would choose formula. The only difference between those groups is that the group that chose formula was more likely to be on public insurance than those who chose donor milk. And among those who chose donor milk, they felt it was more natural and that formula seemed more synthetic, whereas those who chose formula, they didn't feel like they knew enough about donor milk to feel comfortable with it, and they just felt like 1 or 2 bottles of formula wouldn't make a difference because they were planning to exclusively breastfeed. But what was interesting is that in both groups, there was this discomfort, like not really understanding the selection process for donors, like who's giving this milk, where does it come from, how do we know it's not tainted, et cetera. They wanted more information about it. So this is important for us to recognize, those of us who are invested in the use of donor milk, that we make sure that everyone's educated. And I have found this as a family doctor in my breastfeeding medicine clinic. When I see dyads where clearly I need to recommend supplementation, there's undiagnosed, insufficient glandular tissue, this baby really needs to be supplemented right away. I'll ask about, would you be interested in donor milk or formula, or talk about the difference? And they have many questions and they kind of scratch their heads and and kind of squint their eyes and wonder, like, what do you mean donor milk? Like, what is this and who does this? And so it's something that we really need to educate the population about. This was a study just among nurses asking about what their views are of donor human milk on the floor for well newborns. And so they took 20 nurses and had different focus groups. So they're all involved in a focus group about this, where they actually just could pretty much freely talk about their thoughts about this. They felt that in general, they lacked knowledge. So similar to parents, they wanted more information about donor milk, and they suggested more education for nursing staff. They also recognized the parents seemed pretty defeated when they had to recommend formula, especially if they were planning to exclusively breastfeed. So they wanted to be sensitive to that, and they recognized that it made more sense that pasteurized donor human milk would be healthier for newborns, and assumed that it would be a safe source. It sounded like a pretty nice option, but one of the major themes was about their concerns regarding workflow. That they were just wondering, like, how much scanning does it involve to actually pull out the donor milk? Like, do they have to scan it, put in all these codes? They don't have to defrost it, whereas with formula, they can just pull it out of the Pyxis. They can give it directly. They don't have to scan it. They don't have to record anything. They don't have to defrost it. So it just makes it a lot easier in those stressful times to, just pull out a bottle of formula, unfortunately. So we're really making it much harder to use donor milk than formula in hospitals. And then I also want to mention that there is evidence of disparities in the use of donor milk for well newborns. This was a study that came from the Pacific Northwest, where they have about, in this one particular hospital, which has about 2,200 deliveries a year. And they looked at 584 charts of mothers who had the intention to breastfeed and whose infants received supplementation. The mothers could choose formula or donor milk, and they had to sign a consent for donor milk, which is the theme, for in all of this literature. We're still talking about consent for donor milk, but not talking about consent for formula when we know that there is this risk, particularly of cosmic allergy and changing the microbiome. So anyway, what they found is that those who were more likely to receive donor milk were those who were newborns of primates, those who receive supplementation during the day. So it's interesting that the work involved in providing donor milk and asking about consents happened more during the day than at night. Those who had private insurance were much more likely to receive donor milk, and if they were white and non-Hispanic, much more likely to receive donor human milk. And if they were English or Spanish speaking, as opposed to other languages, they're more likely to receive donor milk. So there are some, this may be due to ease of communication. It may be due to the time, not trying to wake someone up in the middle of the night and give them lots and lots of education, being sensitive to that. It could also be implicit bias, though, figuring that people who are more educated are more likely to want this, and those who are not as educated, perhaps are of color, are not as likely to breastfeed, and therefore would not be interested in why bother giving it to them. So in conclusion, I want to just summarize and say that we know that donor milk has been shown to be an effective form of supplementation for asymptomatic hypoglycemia among well infants. And this is not the only study that has shown this. There's another study by SENS that I did not show that has also shown this to be an effective strategy for supplementation for asymptomatic hypoglycemia. That use of donor milk can prevent the use of formula for supplementation, which increases the risk of altering the infant's microbiome, the cause milk allergy, and reducing the likelihood of exclusive breastfeeding. Donor milk, when used for postpartum supplementation, is associated with increased risks of exclusive breastfeeding at discharge and at 6 months. And I want to mention too that there was a study that looked at the supplementation with donor milk as to whether or not that would decrease exclusive breastfeeding at discharge, because it's still a supplementation, and there was no effect. Parents and hospital staff would benefit from increased education regarding the use of donor milk for well newborns and the risk of formula supplementation. And there is disparity, like there is in so many other areas of healthcare, that needs to be addressed when we're using donor milk, including implicit bias. Thank you so much for joining me. Any questions at all?
Thank you so much, Anne.
Yeah, and if you got just a couple minutes, Ginny's really quickly going to go through closing, and then we will prioritize questions for Dr. Eglash.
Yeah, perfect. Okay, so thank you again, Dr. Eglash, for all of that information, and for Summer for your portion as well. I do want to give a special thanks again to the NOHE team at UIC for your support in planning and implementing this webinar. And we have our contact information here, just the info@milkbankwgl.org is our main email. We're also going to put Nicole's email in the chat later on. And then we do have a webinar survey. So again, if you could fill that out, we'd really appreciate that. We will take a raffle for those that fill it out, and you will be eligible for a $25 gift certificate and goodie bag. And then, as I mentioned earlier, in case you weren't on the call right when we started, we are submitting an application for CERPs for IBCLE, and then also an application to the ALPP for the credits for the CLCs, ALCs, and ANLCs. Everyone else can get a certificate of attendance. So yes, that brings me to the questions. We've got a couple in the chat and a couple that we received early on in registration. So I'm going to prioritize Dr. Eglash's questions here. So Dr. Eglash, we have a question here. Should newborns in the hospital receive late-term milk as a supplement instead of term milk, regardless of their gestation between 36 and 40 weeks? So this is specific about gestationally batched donor milk.
Oh, yeah. So I think it's just a matter of prioritization. I mean, I think that it would be ideal if they could get the first month's milk because it's higher in protein. But if we're just supplementing and they're largely taking mother's own milk, I don't think it really matters that much. We really, I think, and I would defer to Summer on this, but it seems like we should really defer that really super precious high protein early milk, preterm, especially preterm milk to preterm, neonates. Unless Summer, if you have any comments on that.
No, I agree. Yeah, definitely. Especially for short-term supplementation for term newborns or late-term, preterm newborns. I think any, early term or the term milk are both great products because they're breastfeeding. So
yes, exactly. Yeah. And my guess in that study, they didn't say specifically, I doubt that, with donor milk, I doubt that they even knew whether it was early term, within the first month. I think that was probably just, most milk that comes in, right, Summer, is from moms who it's not expressed within the first month.
Right. Yeah. The most of the milk supply is term milk. So we do restrict the preterm milk for level 3-4 NICUs, and then the early term for level 2 and 3-4 NICUs.
Thank you. The next question, which was asked, for a mother whose milk supply comes in late and who doesn't have access to donor milk, or she has access at the hospital but not at home, she'll be using formula at home, will her own milk repair her baby's microbiome once the baby is able to get on all breast milk diet again? And if so, how long would that take?
So that was the information from Megan Azad. So Megan Azad was able to show when she did this study showing just that one bottle having a difference in the microbiome, that stool was collected at 3 to 4 months. So that one bottle actually had an effect at 3 to 4 months. So there won't be a complete reversal. So this is a dose related thing, of course. So the more breast milk, the better, and the more that we can limit formula supplementation early, the better. Fortunately, her study showed that just receiving that one bottle didn't have an impact on obesity, which was good. So I don't know that there's a complete reversal once you add in another form of feeding, an artificial milk.
Thank you. All right, the next question for you here is, what strategies would you suggest for anticipatory guidance prenatally to promote an exclusive human milk diet when it's anticipated that an infant will be at risk for hypoglycemia or a delay in lactogenesis 2, like multiple gestational diabetes, PCOS, etc.?
Yeah. Well, I think there needs to be a realistic conversation. So this is it's probably something that I counsel patients on probably 10% or more of my breastfeeding medicine practice when we know that there was a history of low milk production the first time, or they come to me prenatally, know that they're at risk and they wanted to have exclusive breastfeeding. So of course, the first thing is getting off to a good start, right? Making sure that they're skin to skin right away. And that first day, if the baby doesn't nurse well, to express colostrum on a regular basis, giving this baby as much of one's own milk as possible. And then bringing donor milk to the hospital if the hospital is not providing donor milk on the floor. This is something that we did at our hospital. So one of the great things about our milk bank is that we have our dispensaries where people can go to the pharmacy and get donor milk. And this is something that we did at that many families did at our hospital for years, is they brought in donor milk to the hospital. And of course, if it has a label on it, hospitals are usually fine with it. If they know that it's a pasteurized product from, from our milk bank, they would bring that in and use that for supplementation. There's also the DAME study that has demonstrated the expression of colostrum prenatally that can be used, and that colostrum can be used for that early supplementation in cases of hypoglycemia or delay in lactation. But many of those individuals who have like polycystic ovarian syndrome, prone to preeclampsia, who have gestational diabetes, they are at risk for having low colostrum as well. So if you look at the DAME study on how much milk was actually expressed prenatally, like from 36 to 40 weeks, and for some of these moms, it was very, very low. So it's a great option for sure. It has been shown to be safe in low risk pregnancies, and it promotes exclusive breastfeeding. But I can't say that would preclude the need for donor human milk early in those first couple of days.
Thank you. And do you have time for one more?
Yeah.
Okay, so the last one, how would you differentiate between low supply, less than a week old? In other words, how do you know a baby actually needs to be sent home with formula? Healthy nursing behavior is frequently interpreted as insufficient supply.
Well, you don't know. I mean, so, it's all about cadence. Like, it's all about like the change in weight every day. And it's all about like what's on the horizon. So in these situations, you can't, no one has a crystal ball. Someone who has a lot of colostrum leakage during their pregnancy, that is not at all a crystal ball for how much milk they're going to have later. And so they just need to be followed every day. So if they're sent home with formula on day 2, that would mean that someone made a decision already that they need to supplement. So there's something going on right then. And it may not be that they're going to have low milk production over time, but there may be a delay in secretory activation. It may also be because mom received meds. It could be that she has retained placental fragment. There's so many dynamic things that are happening that my personal bias would be that if someone needs to be supplemented, if there's a feeling that the baby needs to be supplemented at the time of hospital discharge, someone who's knowledgeable needs to see them the next day. I really don't advise sending them to a provider who's not very knowledgeable, because there was already like a specialty issue here. Like so many other things in medicine, if someone has heart failure at the time of hospital discharge, or they're, whatever, heart issue it is, and they need like special medication, they need to follow up with someone who knows how to manage that problem. And it may be a cardiologist. And so this family needs to go to someone who can really work with them and understand how to tease out these issues. It takes a lot of sleuthing to understand, why do you need supplementation? What's going on? And no one, I cannot say to someone, in 2 days, your milk supply is going to be fine. Like, I can never say that. And I've been doing this for 30 years. They need to be followed every day. So I would hope that if there's no obvious reason for low milk production, that they just perhaps have a delay in lactation, and that hopefully they're being sent home with donor milk as opposed to formula. Because again, if the intention is to exclusively breastfeed, we want to protect that microbiome. And I love that slide that you had, Summer, about that, the similarity in the microbiome between receiving donor milk and mother's own milk is huge, just huge.
Great. Thank you so much, Dr. Eglash.
Yeah, I'm sorry I have to run off. Thank you so much.
We understand. Thank you so much. And we'll continue with questions with Summer.
Thank you.
Thanks, Dr. Eglash.
Yeah, bye.
Thank you. All right, Summer, we know that we talked about babies outside the NICU receiving donor milk. We do have a question about vulnerable populations that would also benefit from donor milk. Can you quickly give some indications for those other infants?
Oh, wow, there's so many. So we really need to tap into the biological model to think about all the different conditions, symptoms, issues where the baby may benefit from the use of donor milk. The thing that jumps right to the front of my mind is formula intolerance. This can manifest in so many different ways during the first year of life. I've seen babies exhibit formula intolerance very early on. Maybe mom was breastfeeding for a week or two, and then the child's transitioned to formula and just has significant feeding intolerance, where the child has to be hospitalized. So I think that a lot of us who have worked in children's hospitals have seen admissions coming into the children's hospital with severe formula intolerance or feeding aversions or any number of GI diagnoses. So formula intolerance, which can happen early, late, at any time during that first year, and brought up some information about the allergies. There is also, I believe a case for using donor milk for children with NAS or neonatal abstinence syndrome if they're having GI symptoms. So if you think about the NAS scoring system that we use right now, there's a whole section for GI symptoms, weight loss, vomiting, other GI distress. If the child is having significant issues with that particular score, then a trial of donor milk may be warranted and you can see if it helps. So I like to think about donor milk as a trial. You can see if it helps, and if it helps, that's fantastic. We also use donor milk for children with spinal muscular atrophy due to significant reflux and lack of peristalsis. It could also be used for other motor neuron diseases. Donor milk can be used for really any indication, any type of feeding in the newborn period. We also provide donor milk for non-medical necessity cases for families who adopt or for families who use surrogates. They may want to give 1 bottle of donor milk a day just to give some of those growth factors, cytokines, hormones, secretory IGA, and human milk oligosaccharides that we talked about earlier. So anytime a baby's hungry, donor milk is an indication for use.
Thank you. All right, my next question here, somebody had asked how we can encourage families who say they don't want to use milk from someone they don't know. And I know Dr. Eglash presented about the one study about this, and she talked about education. Is there anything else that you would want to add to that question?
Yeah, I think 2 things help with that discussion. I feel like the analogy to blood banking is very helpful for some families and for some medical providers who maybe are unsure about donor milk or using donor milk from another mom. So the blood banking analogy is very helpful to say, well, we, already have, blood banking, and sometimes babies need to receive blood from another person. So this is similar to that. And another strategy is to emphasize how safe donor milk is, to explain that it's pasteurized the same way that some cow's milk is pasteurized in the grocery store. So using analogies to blood banking or to pasteurization within even the dairy industry, I think is helpful to convey the safety. So those 2 things, I think, but all the nurses and lactation consultants and community providers that are on the call right now probably have a ton of really great ideas that they have been using for years to help counsel families about the safety and the efficacy of donor milk. So feel free to chat any other ideas you guys have for how to talk to families about using donor milk.
Thank you. And this is a question that we generally may provide many slides about, but how safe is donor milk? Can we narrow that down in a quick answer, Summer?
Yeah, I mean, just really briefly, the Human Milk Banking Association of North America, who is our accrediting body, I guess to give an analogy similar to the Joint Commission how they accredit hospitals or DMV. So within the Himbana system, which was founded in 1985, milk banks that use that system and who are accredited through Himbana, to our knowledge, there has been no instance of disease transmission or any harm that has come to an infant by using donor milk. In that same time period, several infants have been harmed by formula use, including death due to contamination. So it's heartbreaking to think about, contamination and things that can happen when formula or really any food or beverage are used for infants and for adults. I'm a food safety scientist, so that's kind of my focus is food safety and foodborne illness. So I'm happy to report that our pasteurization process and the processes that we use and all of the regulation that we have here at the Milk Bank maintains an extremely safe framework for milk banking that's all based on food safety and global food safety mechanisms like the HACCP system and the Food Safety Modernization Act. So how much time do we have? Because I could talk about food safety for a couple hours here, but what I'm trying to convey is pasteurized donor human milk is incredibly safe.
Thank you. And kind of along that line, somebody asked a question about what is screened for specifically, and in particular, is coronavirus included in the screening?
Oh yeah, that's a great question. So I do have a couple of comments about COVID. So fortunately, we don't need to screen for SARS-CoV-2, which is the virus that causes COVID-19, because it is not transmitted in milk. And even if it was, the pasteurization process that we use completely inactivates that particular virus. And that is based on research, a lot of research, but Unger's research from 2020, a research group up in Canada demonstrated that SARS-CoV-2 is totally inactivated with Holder pasteurization, which is the process that we use. So we actually don't screen for COVID-19. We do screen for many other types of lifestyle choices, disease history, and health history. We do blood tests for 5 communicable diseases. So we blood test for HIV, hepatitis B, hepatitis C, syphilis, and HTLV. And then pasteurization destroys all vegetative bacterial cells. We also pre-test for bacteria before pasteurization and after pasteurization. And we also drug test the milk for 6 common drugs of abuse here at Milk Bank WGL. So we have an incredible safety system. We have our own internal microbiology lab where we test the milk. So we're all about testing and safety here.
Great, thank you. We did have several questions about costs for hospitals, including startup costs and specific questions about how much it would cost for certain amounts of patient volumes. I'm not going to pass this question to Summer right now. There's so many variables in providing donor milk at hospitals and also through dispensaries. So I'd like to ask if any of you are interested in more specifics about your hospital or program, if you could email us. I'll put Nicole's email in the chat. Nicole Geringer is our education manager, and she can talk with you more specifically about your specific case and questions, and we can help you understand some of what those information would be. And then I do have a couple questions about insurance coverage. Summer, will public insurance cover donor milk for non-NICU babies? Yeah,
so that is state by state right now. We are fortunate that half of our region that we serve, so we are the Wisconsin-Illinois Milk Bank, so we only serve 2 states. And right now there is coverage in Illinois. We are pursuing coverage in Wisconsin too. So all my Wisconsin friends and family that are listening right now, we are coming to Wisconsin and to lobby and to help advocate for donor milk coverage in Wisconsin. Right now, donor milk is covered through 6 months in the state of Illinois for certain medical indications and through 12 months for additional medical indications. So for the first year, if the child has significant medical need, we may be able to get donor milk coverage, and that's both through Medicaid and through private insurance. There are many loopholes, as you all know, working in the medical field. So we work individually with every single patient and with their insurance company. And Ginny is our insurance queen. So thank you, Ginny, for enrolling us with all the different insurance agencies and contracting so we can help serve families in our region. I will just say off the top of my head, I do have my state Medicaid, the state insurance flyer up there. And I believe it's also on our website. So if you go to our website, I believe you can find information about that as well. Hypoglycemia is listed, as well as congenital heart disease, history of neck or risk of neck, low birth weight, less than 1,500 grams, and that can extend past the NICU hospital stay. We often care for NICU graduates who leave the NICU with prolonged feeding issues, and we're able to provide donor milk that is billed to the insurance company for that. So so check out the website and you can see the list of indications through the state of Illinois. We'll be advocating for similar coverage in the state of Wisconsin.
And Summer, to piggyback on that, for those that are not covered by insurance, is that then paid out of pocket by families?
Yes, we do also offer charity care. So we grant about $100,000 a year to families who cannot afford donor milk. And the amount that we grant is based on need, and it's also based on medical reasons. So if a child has a significant need for milk, and we know that milk will keep the child out of the hospital, or if the child's on hospice, then we provide significant charity care, if not, 100% charity care, depending on the case. We also provide 100% charity care for a short-term basis for all cases of maternal death in Illinois and Wisconsin. So if there's any questions about insurance coverage, charity care, especially if there's medical need, please contact us and we will work directly with the family and the providers to make sure that we can help out. That's why we exist. That's why we have a nonprofit here in Wisconsin and Illinois, is to do all of these things that we're doing. We partner with the hospitals to save healthcare costs. We also partner with families to provide life-saving donor milk.
So it's just about 12:30 here. I do have about 5 more questions. So we can either ask a few more questions for those who want to stay on, or we can send some answers in emails. Summer, do you have any time consideration?
Oh, I'm happy to stay on. Yeah,
okay.
Yeah, you guys know I love talking about donor milk and biology of human milk. So if that's okay with Marjorie.
Yes, all good with me. Thanks for checking.
Okay, so I have a good allergen one for you. For older toddlers who continue to nurse at the breast, how does breastfeeding protect for the immune response once they're consuming table foods? Are they at increased risk for allergies?
No, I think that it's been shown multiple times that human milk helps mitigate or negate allergic risk and atopic disease. So there's a lot of different theories. I think the most common or well understood theory is kind of going back to the hygiene hypothesis or exposure hypothesis. So when babies are receiving breast milk, they're receiving a lot of antigens from mom's diet and they're becoming used to that. They're developing a tolerance to those antigens. Peanuts is a great example. So years ago, we had kind of bad advice to avoid peanut products for the first year or two of life, because we thought that that would help prevent allergy. We found out it was doing the opposite. So failure to expose children to peanut antigen actually led to a higher incidence of peanut allergies. So if you think about that and the very strong evidence for that, you can kind of make sense of the mechanisms behind milk. Not only are you providing some of those antigens to build tolerance and exposure, but there's all sorts of different signals. Remember the cell signaling that we talked about? So the hormones, the growth factors, and the cytokines in human milk are communicating with the baby cells, promoting that immune development and tolerance of antigens. So I believe that protection goes all the way through toddlerhood. There's no end time for that, and the benefits would continue.
All right, great, thank you. I have somebody that asked why there aren't more donor milk programs around the city. So if you could talk a little bit about what we do have and maybe where some of our areas for growth are.
Yeah, absolutely. So we do operate depots, which are drop-off sites for donors. and dispensaries where families can go pick up milk all throughout Wisconsin and Illinois. And we've been around for 5 years. So we're a fairly new milk bank in the big scheme of things, and we're constantly developing and growing. We just opened 3 new sites in the city of Chicago in the past month or so. So we're rapidly growing and developing. So if you are interested in developing a new depot or a new dispensary, please reach out. In addition, we want to be very mindful and thoughtful about where and how we're opening these up. So we're really seeking community engagement to make sure that this is a collaboration and a partnership with the community. And that is through our community relations program and our equity program that is run by Titina Sanders Bay. And our crew here at the milk bank. So we are very thoughtful with how we open depots and dispensaries, and really depend on community groups and all of you to help us navigate through the best and the most appropriate places to open up new locations and to identify where there's need. So thank you to everyone on the call right now who either runs one of our depots or dispensaries, or who advocates for donor milk use. We appreciate all of you. And to the donors, I saw, due to HIPAA, I can't say who she is, but she is a nurse from Wisconsin, and she's one of our donors too. So thank you to all of our milk donor superheroes who donate milk to save tiny lives. We appreciate all of you.
Well, that brings me to another good question here. What marketing steps are we taking to inform our younger generations to the option of donor milk for eligible infants?
Whoever sent that question, can they call us for a brainstorming session? Because we really want to do more outreach with young people. We recognize that's an area for improvement within our organization. We want to represent everyone that we serve throughout our community. So I think that that's homework for us, is how to connect more. We just started our Instagram account not too long ago. So we hire a lot of young people here at the milk bank. Some of the young people are still here working with us, helping us grow. Some have went on to other graduate programs, and some are even went to nursing school. So definitely the people that come in and help throughout the milk bank help us with that young perspective, but we can definitely grow in this area. So please chat some ideas, or contact us, or email us with ideas on how to connect with the younger people, so that everyone knows about their options. Because that's what we're really focused on, is just providing education and letting families know what their options are. And we support every family's feeding decisions.
Thank you. This is a nice, easy question. Would we be willing to do a presentation for the Milwaukee area?
Of course, we love Milwaukee. We're going to wear green because we have to celebrate the Bucks winning the NBA championship. So when we go up to present in Milwaukee, we'll all be wearing green to help celebrate. And I do have the Milwaukee skyline as well. Look at that beautiful city on the lakefront. So we love Milwaukee, and we go up there all the time. So we will be there to present. We'll Zoom, and we'll see you at Summerfest.
All right. Let's see. A mom that somebody is working with believes her baby is reacting to dairy and other antigens in her milk through a process of elimination. Do you know of any need for a specialized screening for those who may have babies with similar allergies?
So I think I understand the question. It can go in a couple different ways. If we're talking about donor milk, if a child does have a significant allergy to cow's milk protein or another allergen, donor milk may also contain those antigens. So that's something to keep in mind. Donor milk may be used under medical supervision as a pilot, but if a child has a very significant allergy, I would defer to the physician and the allergist who are caring for that child, because donor milk may also have those antigens. If it's a mild case, perhaps donor milk will help versus formula. But again, I would want a physician or a provider to be engaged with that family for medical supervision. If the question was related to mom's milk, that is fairly common, and elimination diets are great because you can see what the child's reacting to and maybe do some diet modifications to extend breastfeeding with a reintroduction later on under medical supervision. So really complex topic, more common than we think. And so for any families that you guys are working on, with protein sensitivity or with an allergy, engage the physician for some medical supervision, and donor milk may be used as a trial in certain situations. So it is a case-by-case basis.
Thank you. And we have a recommendation in the chat to maybe work with local WIC offices for connecting with younger families. So that's a great idea.
Oh, love that. Love that. We love our WIC partners. Absolutely.
Yep.
Yep.
And then another specific question here, a baby who is exclusively breastfed and has specific food allergies, is there a link to give direction or rationale as to when to reintroduce these specific allergens in mom's diet so as to reintroduce them to the baby?
Yeah, so the Academy of Breastfeeding Medicine has a protocol, allergic proctocolitis, I believe. So I would definitely reference that. Also, I have a little bit of an older algorithm and guidance from a European allergy group. So there are very specific ways to reintroduce. I don't know the guidelines off the top of my head, but I would definitely refer to either the Academy of Breastfeeding Medicine or one of the professional groups, allergy groups, for those recommendations. Off the top of my head, I believe it was you would wait till at least 9 months to reintroduce those foods, but I don't want to give inaccurate information. So Academy of Breastfeeding Medicine would definitely be a great place to start with those guidelines.
All right, thank you. And what's the storage time for pasteurized donor human milk?
Well, the nice thing is that the storage time is a year from the pump date. So we do have an expiration date on the bottle, which will alert the provider or the family when they have to use the milk by. And when the milk is thawed, it can be used for 48 hours. And thawed is a completely liquid state. So once it's thawed in a completely liquid state, you have 48 hours to use the milk. So that helps with resource safeguarding the resource in the hospital and also for families at home.
Great. And this is our last question, I believe. Can you talk a little bit more about calories from donor milk? So I think the question is asking, do we get more higher calories from donor milk, or anything you can share about the caloric content?
Yeah, so here at the milk bank, we batch by gestational age and developmental age of the milk to match it to the baby. So we have preterm, early term, and term milk products. The average calories in all three products is 20 calories per ounce. The main difference is that the protein is higher in the preterm and early term products, with an average of 1.5 grams per deciliter of protein in those products versus about 1 to 1.1 grams per deciliter in the term products. So our donor milk products have excellent nutrition profiles. Part of that is the way that we process and pasteurize the milk. So the macronutrients are largely intact and remain stable throughout pasteurization. It's really some of the bioactive components that decrease with pasteurization, not the nutritional components of fat, calories, or total protein.
And I actually have one more, if you can add one more.
Yep,
All
let's do it.
Has there been any consideration to accepting donor milk from older moms who have breastfed and want to relactate to donate?
Oh, wow, that's a really good question. So right now we do accept milk through the child's second birthday. So we are able to accept milk during those, first 2 years of life. And that is based on research on the composition of human milk through those first 2 years. Right now, we don't have enough research on the composition of milk, due to relactation at a later stage, but that's something that we're always open to. We have very open minds here because we adhere to the biological perspective and the biological model of lactation. So I'll keep my eyes out on that research, and Himbana is really good about improving and updating their guidelines and standards based on new research. So we will definitely keep our eyes out on the relactation research and be open to accepting that milk in the future.
All right, thank you. I think that's all the questions. There were a lot of great questions today, so thank you everybody who stayed on late to listen. I just put the survey link in the chat once more. We'll be sending a follow-up email with some information from the slideshow, the survey link, and to keep you updated about the certification credits. I'm also going to put in the chat a link for our website, both how to donate and how to order milk. And then I have a bonfire link as well. We just got some new swag in our shop for breastfeeding week in August. So there's a new design if you want to get some cool shirts. Summer, anything else to add?
No, thank you so much. Thank you to Nohi. Thank you to Health Connect One, Ginny, Nicole, Dr. Eglash, everyone on the call right now. Thanks for the boots on the ground work that you do supporting moms, helping moms reach their breastfeeding goals. And, you know, we appreciate all of you. So this is a community effort. We're your community milk bank and we're here to serve you.
Thank you.
Bye, everybody. Thank you so much.